Emergency! Evacuate now.
Evacuate via the nearest exit.
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 Emergency!
Good evening, everyone. Welcome back to the 10th week of problem solving session of medicinal chemistry. I'm Gitanjali Singh, your course TA for this course, and I'll be hosting the problem solving session. This session is held every Monday from 6 p.m. to 8 p.m. live on Google Meet. This session is recorded. During the session, we discuss the week summary, last week's assignment, some modular questions based on week's topic, and we try to discuss any queries that you have regarding the topics that have already been discussed. In case of network eruption, please do not leave. Wait for a few minutes. If it's from my end, I'll try to join as soon as possible with the same link. And if for some reason you get out of the Gmeet, you can join with the same session. It's valid for the entire 2 hours. Please post your questions in the chat box during the sessions. But because we don't host that many students, I would advise you to unmute yourself and just ask me the question then and there. If your question requires longer discussion, I would like to request you to be more patient, and then we can discuss the question at the end of the session. And probably if I don't know the answer to the question, I can prepare some more slides and share and try to discuss the question before we start the new session next time. This presentation is recorded and the link will be shared to you along with the slides. Now we'll get back to our session. If you remember, I was requested to have a brief discussion on CRISPR-Cas9. So I've prepared few slides on CRISPR-Cas9. So if you are not interested in CRISPR-Cas9, you can just leave and join later, maybe in 10 or 15 minutes. But I would still suggest that even if you are not interested, try to stay for a few hours, because this is a very interesting and a very good topic, and it never harms to know a little more. So yeah, CRISPR-Cas9. In the late 1980s and early 1990s, researchers including Francisco Mojica, they noticed unusual repetitive DNA sequences in bacteria, prokaryotes. These sequences were short and palindromic. So when you read it from right hand side to left and from left hand side to right, they appear to be in the same manner. These are called palindromes. And these unique sequences that were like filled with spaces, they were known as CRISPR, clustered, regularly interspaced, short palindromic repeats. Hence, the abbreviation CRISPR sequences. The function of this CRISPR were not known at that point. They just know, oh, there is a sequence, and they were curious to know more about it. But for like 20 years, no one exactly knew what were these CRISPRs doing. In early 2000s, Alexander Bolotin and his colleagues identified CRISPR-associated protein called Cas protein, which is known as Cas9, and proposed that its role is to cut DNA in a particular sequence. So they are basically cutting up this DNA. So that's how they came to know about Cas9 protein. In 2007, Philip Horvath and his team demonstrated CRISPR sequence work with Cas9 proteins to provide adaptive immunity against viruses. So these are in bacteria, and this is a immunity thing, like that, defense mechanism that bacteria have against viruses. Now, CRISPR sequence and Cas enzymes work together to incorporate. So how does this actually happen? Like how are bacteria protecting themselves with the help of CRISPR against these viruses? So what happens is, suppose a virus is trying to attack a bacteria. It comes inside it, and we know how viruses replicate. So they are borderline between living and non-living. When they come to when they attack a living organism, they basically direct the um, DNA or RNA of the living organism to replicate. They basically use their genome and their DNA to hack it and do their work. So when bacteria attacks a cell, CRISPR sequences and Cas9 enzymes, they work together. They they get the DNA from the bacteria. They cut it in a this Cas protein. They cuts it in a particular sequence. They acquire the spacer. So they have palindromic repetitive palindromic sequences they and that are separated by spacers. Now this bacteria, with the help of Cas9 protein, and it cuts down the DNA from viral infection and incorporates it as spacer between this palindromic sequence. This allows a bacteria to recognize and defend itself in future. I'll explain how. So the first step is cutting and capture. So when a bacteria are infected by virus, they use this CRISPR system to cut the invading viral DNA. They cut it and incorporate it as spacers in their own genome. So that becomes memory from the viral attack. Now, the second step is monitoring. The bacteria then transcribes spacer DNA into CRISPR RNA. This is known as cRNA. The Cas9 and the cRNA, they form a complex and they monitor for any the DNA sequence complementary to the RNA. Now the third, defense. If the matching viral DNA, the one that they got from the virus infection, is encountered, the cRNA, you remember, the CRISPR RNA and RNA Cas9 complex binds to and cuts the viral DNA to prevent it from replicating. So it has a memory of it, and now it recognizes, oh, again, this is the sequence that I'm getting, and this is the one that caused infection last time. So what it does is, it has everything, it has its weapon ready. Now it cuts the viral DNA from when it comes to the next time. So you can see here in the figure uh, that is shown in the slide, the first one is acquired spacer DNA from viral infection. You can see that there is Cas1 Cas9 Cas2 complex with the bacterial DNA. Now it is added as spacer to genomic CRISPR region. You can see here Cas genes, and this repeats our palindromic sequences. I don't know what's happening with my mouse right now. Okay, and now you again seen this in uh, this image. The the they so next time the virus enters the bacterial cell, the cell is ready with all the weapons, right? The CRISPR RNA, along with cRNA and RNA's Cas9 complex, it recognizes the particular DNA infection that was caused last time, and it proceeds to cleave that, and then that's how it basically prevents the um, new viral infection. So using this system, bacteria can collect sequences from different infecting viruses, and they basically create a library. So remember how this is different sequences, and these sequences basically add on to the spacer, add on as a spacer, and they become memory of the viral infection for the bacteria. And this, it can accumulate over a period of time. So every time there is a viral infection, the bacteria retains a memory from it, and because it's a part of the bacterial genome, it can be transferred from one generation to the other, and they can basically keep on adapting and keep on collecting the memory over the period of time and can transfer and transfer, right? Now, let's see how we utilize this as a gene editing system. So, in 2012, Jennifer Donda and Emmanuel Charpentier, they demonstrated that CRISPR-Cas9 system can be repurposed to edit DNA in living organism, and for that they were given Nobel Prize in 2020. In 2017, so after 2012, in 2017, Feng Zhang and his colleagues demonstrated the use of CRISPR-Cas9 to edit mammalian genomes. This itself is a very, very, you know, path-breaking application of CRISPR-Cas9. So we know that mammalian genome is very complicated, and we have double-stranded DNA and very different from very so much more complex than bacterial genome. And to use this application, to actually use it to edit mammalian genome, it's mind blowing. But yeah, let's discuss how they did that. So the CRISPR-Cas9 system was uses modified components of bacterial CRISPR system to target specific cutting of double-stranded DNA. The DNA repair mechanism then takes over to fix the manner that modifies genetic sequence that has been cut. So the first step is cutting the DNA. The Cas9 enzyme, you know, the Cas9 enzyme, it's basically an endonuclease that cuts both strands of double-stranded DNA at specific site. Now, multiple types of Cas enzymes are found in nature, out of which Cas9 is commonly used in the labs. There's one more thing, the single guide rRNA. So it's an engineered RNA that forms complex with Cas9. This SG single guide RNA, is fusion of two regions that occur as separate RNA in nature. So this is engineered. One is different thing, one is different thing. One is known as the guiding region, the other one is known as the scaffold region. The guiding region is part of CRISPR RNA or crRNA in nature, which is 20 nucleotide region that is complementary to the target region, and that defines the target of DNA sequence that Cas9 cuts. Now, scientists can customize this sequence based on their own requirement for the target. And the next one is scaffold region. This is trans-activating CRISPR RNA that is known as TRA-CRRNA, track CRRNA. Just remember it like that because it's trans-activating. You can just remember as track, and then CR, we know that how we are using CRISPR associated RNA as crRNA in nature. This binds to multiple hairpin, like you can see in this hairpin loop. Okay, so this region forms hairpin multi hairpin loop structure scaffold that binds to the crevice of Cas9 protein. You can see here there are 5 steps of Cas9 DNA cleavage. You can see here. So the Cas9 binds an sgRNA. It recognizes and binds the scaffold, that scaffold that we talked about, that multi-hairpin loop region of sgRNA. The nucleotide sequence of the scaffold region determines its structure, which is tailored to fit within the Cas9 protein as key fit into the lock. So remember how we talked about the complementary sequence? This is like very well engineered way of directing the Cas9 to cut a particular. There is photospacer adjacent motif known as PAM. PAM. It is required for Cas9 function. This sequence motif is immediately downstream of the target sequence. Now, Cas9 recognizes recognizes PAM sequence 5-NGG, where N stands for any nucleotide. It can be A, T, G, C, whatever. Now, when Cas9 binds to this photospacer adjacent motif, PAM, you can see here, it separates DNA strand to the adjacent sequence and allows the binding of sgRNA. So, if sgRNA is complementary to the sequence, the Cas9 cuts the DNA. Now, step 2 is repairing the break to engineer the change. Now, researchers can use their own cell DNA repairing mechanism to modify, delete, or insert a new nucleotide sequence. The repair can happen in two ways. One, non-homologous end joining, that is known as NHEJ. So the enzymes reconnect ends of the DNA break, remember how they were cut, and this process can randomly insert or delete one or more bases, and this can cause, because this is random, this can cause mutation and can disrupt gene function or its expression, because obviously because it's random, so it might delete one base pair, two base pair, and that's how we know there is point mutation, frame shift mutation, whatever. So this can basically disrupt gene function and expression. The second way is homology directed repair, known as HDR. The proteins patch up the they basically patch using donor template DNA. This is designed particularly by scientists and it's flanked on both sides of homology arm. The match that they basically match the sequence upstream and downstream of the cut, and a complementary DNA strand is created during the repair. So you can see here the SGR. We talked about how it was cutting, in the last one, and the second step is Cas9 requires a particular PAM sequence that we discussed to present directly adjacent to the photospacer sequence. You can see here PAM. When the Cas9 sgRNA, that single guide RNA complex recognizes and binds with PAM, it separates the DNA strands of the adjacent sequence and allows the binding of sgRNA. Now, the guiding region of sgRNA binds to the target DNA sequence. The guiding region of the sgRNA attempts to base pair because it's complementary with the DNA. If the match is found, the process continues. Otherwise, if the complex is realized, okay, a match nahi mil raha hai, so otherwise the complex releases and attempts to bind to another PAM and target DNA sequence. And the fourth one is Cas9 makes double-stranded DNA break in DNA 3 base pair upstream of the PAM. That's how they're doing it. And in fifth step, the Cas9 sgRNA complex releases the cut DNA and is ready to repeat the process again. So that's how we use CRISPR-Cas9 for editing gene. Now I can imagine how this is very complex and it seems like a very difficult thing to follow through, but I'll try to explain this in a much more simpler way, not with technical things, but overall just to get a more clear idea of what CRISPR-Cas9 is actually, just to get to the base of it. So it's basically a tool that so what is CRISPR-Cas9 in very simple layman terms? Let's discuss what it is. It is a tool that scientists use to make changes, also known as edits. So when I talk about edits, it's basically changing to the DNA of living organisms. This living organisms can be plants, animals, humans, anything. So it's like a pair of molecular scissors that we talked about that can cut DNA at specific spots, allowing the scientists to remove that particular spot and maybe join it back or add something, or basically just delete or add. So the basic idea is, imagine DNA as long instruction manual that tells the cell how to behave, like how to build and how to operate. Sometimes there are mistakes like mutations, in manual, and that causes disease. So sometimes due to deletion of some genes there is a disease, or maybe due to extra pair. You understand, right? Now the CRISPR-Cas9 can allow scientists to find a fix, to you know undo this mistakes, to correct this mistakes by cutting and editing the DNA at very specific location, almost like fixing a typo in a book. Now, how does CRISPR-Cas9 work, you might ask? So, CRISPR, as I said, is clustered, regularly interspaced, short palindromic repeats. It is a special part of DNA in bacteria. Um, bacteria use this to remember the viruses, as we talked. They remember how viruses that attacked them in the past. When a virus tries to attack again, the bacteria can recognize it and cut the virus DNA using a part of CRISPR system. So in lab, the scientists can use this memory of like previous attack from the uh, how to explain. So this bacteria, they have previous memory of the viral infection, and they cut that part of a genome and add it to their spacer, between the repeats. They added it as a spacer. Now they can so now we have this bacteria who has the memory of the viral infection. Scientists can basically use this to guide Cas9 protein to exact place in the DNA that they want to edit. Now Cas9 is CRISPR associated protein 9. Cas9 is a protein that acts like molecular Caesar. It can cut DNA at specific spot, but it needs some kind of guidance to know where to cut from. This guidance comes in small piece of RNA that matches the DNA sequence scientists want to edit. Okay, I hope that's clear till now. Now the RNA is like GPS. This RNA is like GPS that directs Cas9 protein to the exact location. We discussed the steps, which was quite complicated, and I saw you guys leaving, but I'll try to explain it in a more simple term. So the first one was uh, guide basically cutting off the DNA, editing. So guide RNA. So scientists create a small sequence of RNA that matches with a specific DNA sequence that they want to change. This RNA, wait. Hi, Kritika. I see you have raised your hand. You can unmute yourself, and is there anything you'd like to ask? Kritika? So I was requested. Oh God, I'm so sorry. I've been doing this entire time by muting myself. Oh, I'm so sorry. I'm going to repeat this again, like the simple form of CRISPR. It's so embarrassing. I'm so sorry. So, yeah. Yeah, thank you for reminding me to unmute myself. Okay. So, we are discussing uh, CRISPR-Cas9. As you might remember, I was requested to briefly discuss what CRISPR-Cas9 is and how do we use it as editing tool. So I've explained this in a very technical term, and it's I've written basically everything in the slides. So would you like me to repeat the technical terms, or you just want to get a basic idea of how things are, and I'll repeat it in a very easy to understand way? Or do you want me to explain this in a very technical terms? Just let me know. Since you're the only one, I can do it again. What do you want, Kritika? I'll try to give you a basic idea of what CRISPR-Cas9 is. And okay, perfect. Yeah, because once you get the basic idea, you can read about it and understand more, and probably get a better understanding of everything that's happening in the slides. I'll repeat. So basically, CRISPR-Cas9 is a tool that scientists use to make changes to a DNA of living organism. This living organism can be plant, animal, or human, whatever. So it's basically, you know, using molecular scissors to cut DNA at very specific spots, allowing the scientists to um, remove or add particular sequences to the genetic material. Now, basic idea is, imagine you have a DNA that tells the cell how to behave, how to build, and how to operate. Right? That's what DNA does. It basically helps transcribe the protein. That protein helps us do daily things, like, for the cell to build, normal metabolic processes, everything, to basically guide enzymes. So yeah, DNA basically gives the manual, is an instruction manual for telling the cells how to build and operate. Now sometimes there is certain mutation, and due to this mutation in particular sequence of DNA, there are some diseases that are caused, some due to point mutation, frame shift mutation, deletion, things like that. There arises mutation, and this mutation leads to an irregular activity in enzyme function that somehow affects the normal metabolic process of the body, and we get some kind of disease. Now, using this CRISPR-Cas9, scientists can basically find a fix to correct this mistake. They can basically go and cut the sequence that is mutated or that has some kind of mistakes. They can cut that precisely at that spot. They can basically remove that sequence that is not working properly, that's not coding properly, or maybe have a good sequence, like the correct form of the sequence, and add there. So that's how they use this CRISPR-Cas9 technique to edit the DNA at a very specific location. That's basically like correcting typo in a book. Okay, how does this work, you might ask? So CRISPR is clustered, regularly interspaced, short palindromic repeats. So this is special repeats of uh, DNA sequences in bacteria. I'll show you here. So that are repeated in a regular interval. So when you read it from right to left or from left to right, they're the same sequence, and they have in them what's called spacer. So suppose you're A, right? And then there is space. Again, there is some other palindromic sequence, like suppose CCTTGGCC, something like that, like kayak or ata ya tatat box, something like that. Okay, so important thing to remember is there is spacer that's in between this palindromic sequence. Now, this is the special part of DNA in bacteria. Bacteria use this to remember the viruses that attacked them from before. So I'll explain it from this slide here. You can see there is a bacteria trying to enter the bacterial cell. So what the bacteria does is it cuts the DNA of this virus in particular sequence and adds it as spacer between this palindromes, between these sequences. And now the bacteria has memory of the gene of memory of the sequence of the viral DNA. Okay, and it has become part of its genome. So next time the virus tries to attack the bacteria, it remembers that it has the spacer, the sequence, the sequence of DNA, it matches that DNA with the new virus infection. It recognizes, oh, this matches with this new virus, so this is the virus that infected us last time. And it goes there with its CRISPR-Cas9 and cuts it in the particular sequence, and hence it saves itself from the new infection. Okay, this is very lame, very basic term of understanding what bacteria is doing with this CRISPR-Cas9 thing. And because this becomes part of their genome, you know, so it can keep on adding. So suppose this was the case of one virus, next time another virus come, the bacteria does the same thing with it, and it keeps on adding to its library. Now this is from last infection, this is from this infection, and that's how it keeps memory of the new infections. And because it's part of their genome, it can transfer that information from one generation to the next generation and keep on adding and adapting the library. So yeah, that's it. In in labs, what scientists do is they use this memory to guide Cas9 protein to the exact place where they want the DNA, and to guide the DNA, and where they want to edit the particular sequence of DNA. Cas9 is CRISPR associated protein 9. So it's a protein that acts like molecular scissor and can cut DNA at specific spot, but it needs the guidance to know where to cut. So it can cut it in a particular sequence, but how would it know where to cut, right? So that's where a small piece of RNA comes. We call it sgRNA. So single guide rRNA. It's here. Single guide ssRNA, and it's basically that its sequence has that particular match for the DNA sequence that the scientists want to edit. Now, this particular RNA acts as GPS to guide Cas9 protein to exact location. Now, let's discuss about the steps. So, the guide rRNA, sgRNA, the scientists create small sequence of rRNA that matches specific DNA sequence that they want to cut. They basically that's how they recognize the DNA, and this RNA helps in binding the it gets it guides the RNA to attach to the target DNA sequence because it's complementary to that DNA sequence that scientists want to edit. After binding, Cas9 comes there with its scissor, obviously, because it's a type of endonuclease. Uh, it goes there, it cuts the DNA at the location where the guide RNA has led it, and after cutting they start repairing. After the DNA is cut, the cell tries to repair the break, and scientists can use this repair process to either delete the faulty gene or insert the desired or new corrected form of gene, or maybe change the sequence entirely. Okay, so this is how they do this kind of editing precisely. Before this technique, they didn't have anything as precise and as accurate. It was so hard to edit a DNA, and the accuracy was very bad. With this, they have very high precision, very good accuracy, and yeah, it's much more simpler this way. And the speed, it's very fast, and the cost is much more cheaper because you're not relying on random things, right? So basically, yeah, that's what CRISPR Cas9 is. I hope I could explain it in a simpler terms. So let me go through the steps again. You can see here the Cas9 binds to sgRNA. This is single guide rRNA. The Cas9 recognizes and binds the scaffold region to sRNA It forms a complex with sRNA and Cas9. They form a complex together. The nucleotide sequence of the scaffold determines its structure, which is tailored to fit within Cas9 protein as a key to fit its lock. Like, because it's complementary, so that's how you get the lock and key. This can only fit this one because it's complementary. The Cas9 sgRNA complex binds to PAM site on the target region. This PAM is photospacer adjacent motif. It's basically a sequence just adjacent to the target DNA. This can be any. It's run from five. It's read from 5' end with NGG, where N can be any nucleotide, ATGC, whatever. Okay, so it recognizes this complex of Cas9 sgRNA, recognizes and binds with PAM, and it separates DNA strands, allowing the binding of sgRNA to it. In third step, the guiding region of sRNA attempts to base pair with DNA. If the match is found, the process continues. Otherwise, the complex releases and tries to find the next PAM, just next to the target DNA sequence. The Cas9 now makes double-stranded break in the DNA, three base pairs upstream to PAM, and in fifth and the final step, the sgRNA complex releases the DNA and it's ready to repeat the process all over again. So yeah, that is CRISPR-Cas9. I know it's very difficult to follow through, but I hope with the basic explanation you somehow get the idea of what CRISPR-Cas9 is, right? So let's discuss last week's assignment. I know the answers have been released. So we will discuss why those answers were correct, and that's how we'll try to revisit all the concept that we have previously discussed until week 9. Okay, so the first question of the assignment was, which of the following yeah, Kritika, you can if you have any question, or if you still want me to explain something, please unmute yourself and ask the question clearly, or maybe just write on the chat box and I'll try to answer it. So yeah, let's proceed with the questions. Which of the following will not be a limitation of a pro-drug design? A. Formation of toxic products. B. An inert carrier can be cleaved off, forming toxic product. Sorry. 3. Product cleaving off before reaching the target site. And number 4, the cleaving off carrier is also site specific. So they are asking about what will not be a limitation of prodrug. So the answer was the fourth one. The cleaving off carrier is also site specific. This is basically what essentially prodrug design is targeted to do. So that is the advantage, that is the main point of designing a prodrug. Okay, all the other 3 options are actual limitations associated with prodrug. Now, yes, Kritika. So let's try to understand the prodrug design. A prodrug is an inactive or less active form of a drug that gets metabolized in a body to release active drug at the site of its action. So the purpose of designing a prodrug is to improve bioavailability, to enhance drug solubility and stability, to reduce toxicity and side effect, and 4, to improve the drug targeting. That's why we want the drug to be inactive throughout its way and just to be active at its site of action. That's the whole point of designing a prodrug. But there are several limitations. The first one is formation of toxic product. So the main concern is what if there is metabolite? So the drug the drug that we have designed is active, and we have tested the drug for all kind of toxicity and everything. By making product, by basically conjugating it with something and making it inactive, we are designing it uh, we're we are designing a less active form of it, but we don't want that inactive form to be permanent. We want it to be temporary until it reaches the site of its action. Once it reaches there, what we want is that thing, the modification that we did to the drug, to basically release, and we only want the action of the drug there, not the uh, not whatever was conjugated to it, whatever modification we did. So we want the product of the hydrolysis, or the product of you know basically disconjugation, to be inactive. But sometimes what happens is this leads to formation of toxic metabolites after cleavage of the prodrug. And if the inactive carrier breaks down into toxic products, there can be certain unwanted side effects. For example, in case of cyclophosphamide, it generates acrolein, which causes bladder toxicity, also known as hemorrhagic cystitis. So yeah, this is one of the limitation of having a prodrug. The second option was an inert carrier can be cleaved off, forming a toxic product. So some prodrugs, they have carriers or linkers that are cleaved off after administration. So if the carrier itself is toxic, it can pose risk to the patients. And that's why we try to join some you know try to conjugate it with something that's less toxic. Or even after that kind of, that's why we don't use methanol in formulation, because we know methanol is toxic to cells, and these kind of things can add additional toxicity that is unwanted. So we test drugs for everything, and unknowingly, if we add something that's toxic to help carry the drug from one place to its after administration to its site of action, or maybe near the target, what if it is toxic? So that would cause more problem that what it's supposed to solve. There are certain ester based prodrugs that release toxic byproducts after hydrolysis. So yeah, this is also not the correct answer. The last one was product leaving off before reaching the target site. So we want the prodrug to just be activated near the site of action, or near the target, so that it has some stability and more bioavailability and less side effects. But what if it cleaves prematurely? It will start getting metabolized too early, and by the time it reaches the target, it's you know already in less efficient form, less bioavailable. That basically reduces the sorry sorry that basically reduces the efficacy of the drugs. So early activation of doxorubicin prodrug can lead to systemic toxicity instead of localization action in tumors. So this is one of the example. That's why premature cleavage is also a limitation, and hence not a correct answer. The fourth option was the cleaving of the carrier is also site specific. This is the actual reason the prodrug are designed for, right? This uh, so if they get cleaved at their target site, they become more selective, and they don't have many side effects. So that's the whole purpose of designing a prodrug. Antibody drug conjugates, known as ADCs, they use enzymatic cleavage to activate drug only in cancer cells. Hence, they minimize systemic toxicity. This site-specific cleavage is beneficial and not a limitation, and that's why this one was the right answer. Okay, let's move on to the next question. The next question was D1927 contains a branched alkyl group as shown with blue color, which protects the terminal peptide bond from hydrolysis. What is a general term given for this group, for a group such as this? So, addition of this group, what is it called, right? The option was electronic shield.
Bulkhead group.
Sorry?
This kind of tertiary group is present, then will be calling as bulkier thing which causes steric.
Yeah, perfect. Very good. Yeah, that's the correct answer. The options they're given: alkyl shield, electronic shield, and steric shield. The most appropriate one among this is steric shield because that's the purpose of this group. Though it's alkyl, it's alkyl can be, it's not very specific. That's why we don't call it alkyl shield. Alkyl can be methyl, you know, ethyl, propyl, isopropyl, or anything. This is not very specific. So when we try to answer these questions, we look for the most suitable answer. And the most suitable answer, as you said, is the third one, steric shield. We'll briefly discuss what these four options are. So the option was electronic shield. An electronic shield simply implies that the group that influences electron density around the reactive site. So they were talking about the peptide, and so electronic shield is basically addition of a functional group that or addition of anything that affects the electron density around the reactive site, maybe around the amine or anything, electron withdrawing group or whatever, that makes a bond less susceptible for reactions like hydrolysis or basically cleavage, whatever, by changing the electronic properties of the reactive site. This is not the case because we are physically blocking the site. The second option, stereo electronic shield, it basically is mix up of both the terms, steric shield and electronic shield. So addition of a bulkier group that not only provides a um, physical blockage but also affects the electronic density around the um, functional group. Now, alkyl shield, as we discussed, it's not very specific. It's more or less anything that's alkyl. And we have the option steric shield there, so that's the answer we go for. The next question, salbutamol is an anti-asthmatic agent which shows an improved activity over previous catechol-like compounds. What strategy was used in order to attain the improved duration of action? You can see here, this is salbutamol. It has oxygen, phenol, secondary alcohol, tertiary butyl group. Now, the options given were substituent variation, group shift, chain contraction, and simplification. The correct answer is actually group shift. Let's discuss what the four options are briefly, and then we can um, uh, conclude why group shift is the correct answer and why the others aren't. Wait. So the salbutamol is beta 2 androgenic antagonist and it's used for treatment of asthma as said in the question. So basically this was designed as improved version of catechol like compounds. The catechol like compounds had shorter duration of action and we wanted longer longer duration of medicine, not just one that vanishes quickly. So catechol-like compounds, they had short duration of action because they were easily metabolized by catechol O methyl transferase, COMT. They quickly the COMT quickly methylates the hydroxyl group at the meta position of catechol ring, and this leads to rapid degradation of the group. Now, group shift refers to the strategy of shifting functional group of one molecule, basically from one position to the other, and thereby improving the pharmacokinetic properties. So the hydroxyl group was shifted to a different position from the meta position, and that's how they made it less susceptible to COMT metabolism and increased the duration of its action. This modification, basically improved drug stability and reduced rapid degradation that was seen in the traditional catechol compounds. So the 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 other options. The first one was substituent variation. It refers to the modification at specific side chain or group attached to the core structure of the molecule. You know that the goal of doing this substituent variation is to alter drug activity and other properties like solubility, potency, whatever. And the substituent can basically impart some activity that's not very, you know, very desirable in that original molecule. So that's one thing. That's not, they're basically not changing the substituent, they're shifting the already existing functional group in catechol to a different position. So this is not the best description. Now, chain contraction, we basically shortening the carbon chain length or linker, whatever, and that also changes the pharmacokinetic properties of the molecule, but we see that's not the case with salbutamol. Simplification simply refers to reducing the complexity of the molecule. So after this SAR studies, we we realize the important we motives, the main thing that's imparting activity to the molecule, and we try to get rid of all the extra things that were in the molecule that is not directly affecting the bioactivity of the molecule. So that's called simplification, and that's again not the strategy used in salbutamol. The strategy that led to the improved duration of action is group shift. This involved shifting of a functional group, hydroxyl group, from meta position to the other position and reducing its susceptibility by COMT and allowing the drug to remain active for a longer period of time. Again, I've just tried to add everything that you can refer to. Even if you don't take this class, you can just go through the slides and read them and probably get a better idea of what's happening. So this modification leads to improved in oral bioavailability and its stability and selective beta 2 androgenic receptor activation. So yeah, that's pretty much it. The next question was, megestrol acetate has a short duration of activity since it is metabolized at position 6, placing a methyl group at the 6th position and increasing duration of action. What role does methyl group play? Sorry. The first option is it is susceptible metabolic group. Second, it is a metabolic blocker. Third, it is a conformational blocker. Fourth, it is an electron donating group. They are talking about methyl at 6th position. So what is the addition of a methyl group doing to basically increase the duration of action in this acid?
It is Since it is added to the position where the drug is metabolized, it may be a metabolic blocker.
Very good. Yeah, that's precisely the right answer. And very good. I see you have a very good understanding. That's very nice. So I'll still go through all the options so that whoever goes through this recording, they get a better idea of what things are.
Ma'am, and where do I get this PPT? And you, as you mentioned before.
Oh yeah, so there is a link, recording sheet link, that you might get with the feed when you get the feedback mail for this problem solving session. There are two links, one for the survey and the next one is recording sheet. Just click there and you'll see um, a link for PPT for YouTube channel and everything. Just go there and you'll see you have a log of all the PPTs to my drive. I have saved everything on my drive. You can just go there and find PPTs from week 1 to week 10. As soon as the lectures are done, I upload this PPT to my drive and share the link.
Okay, thank you, ma'am. Ma'am, and one more question.
Yeah.
Like while we are discussing about the questions, at sometimes you are just quoting some the things from a book.
Yeah, yeah.
Do you have any specific book for this thing? I'm totally new to the syllabus,
Yeah,
so I
yeah.
have no idea about the book to refer.
Yeah, sure. So, yeah, there's a book. Uh, I can share the link of the book as well. Next time,
Can I mail it to you, ma'am, so
sorry.
that you can? You have given your mail ID. If I'm able to connect you through mail, you can send me through
Yeah,
that.
yeah, sure. And even if I don't, next time what I'll do is, I'll definitely mail it to you. But sometimes uh, I think the book is larger than 25 MB, so I might not be able to mail it to you directly. But what I'll do is, I'll upload that book to my drive, okay, and I'll share the link next time in the chat box and I'll pin it. So even if you join it for a little while, you can just go through that link and download the book. I have that book downloaded already. So
Okay,
for
ma'am.
that,
Name of the book, ma'am?
it's Patrick G's book. Just you know, just let me just a minute, okay? Let me give you the proper name so that it's easier for you to search the book. So there are two books. One Graham Patrick's book, and the other one is Foy's Medicam 7th edition book. Foy's Medicam, and the other one is Graham Patrick's book. This one is it's more concise and a shorter book, and it's easier to read. Another one is an introduction to medicinal chemistry. This is the book that Sir uses and almost everyone uses. This is 5th edition, Graham Patrick's book. The author is Graham Patrick, and the name of the book is An Introduction to Medicinal Chemistry. So I don't know if you can see that it has interface like this. Okay, so if you find it, well and good. If you don't, I'll upload it to my drive and I'll share the link and you can download it from the link.
Thank you so much, ma'am.
It's okay. Um, One more thing, if you feel like there are some things that you don't understand properly and that I might just you know say it in a flow, you can just stop me there and you can ask me to explain. Most of the time, I might not be able to explain it in a easy to understand way, but I'll try my best. And next time, I'll try to be, next time I can add a few more slides and try to be as conceptual as I can. And probably that will help you clear the concept and understand it in a much more better way, I guess.
Okay, ma'am.
So, the megastrol acetate is a synthetic progestin used for a variety of medicinal applications, sure. And it has short duration of activity because it gets metabolized at sixth position. You saw it in the structure. At this position, the compound is metabolized, mostly via oxidation. Though I've written another, sorry, it's mainly via oxidation, and that reduces its effectiveness. So by adding methyl group at sixth position, they basically reduce the compounds, you know, uh, activate a compound's susceptibility to get metabolized, and it prevents the rapid metabolism, the compounds from being rapidly metabolized, and that's how they increase the duration of drug action. The methyl group acts as metabolic blocker, and does it make and that's how it makes the uh, enzyme difficult to recognize that point and break it down. The strategy of placing a methyl group at a metabolically vulnerable site is often used to increase the stability of a drug in the body and to prolong its activity. By preventing or slowing down the metabolism, the drug remains in circulation, in the systemic circulation, for a longer time, and that's how it retains its therapeutic effect for a longer period of time. Why the other options are not correct? Well, the first one was it is susceptible metabolic group. No, actually, that's not the case because we just discussed why that's the correct answer. A susceptible metabolic group would refer to a functional group that makes a molecule more prone to getting metabolized by the enzymes. In this case, addition of the methyl group is reducing its chance of getting metabolized. So, no, the first option cannot be right. The second one is it is conformational blocker. The conformational blocker is basically a modification that changes the three-dimensional geometry of the compound, and that prevents the receptor binding, or that's basically to inhibit its biological interaction. The methyl group addition to the sixth position does not alter the overall 3D conformation of the molecule, and therefore, it is not a conformational blocker. It is an electron donating group, that basically increases the electron density around the molecule. So when you can say that when it's attached to acid or anything, NO2, something like that, it donates electron via inductive effect, like plus I effect. But in this case, the electron donation is not that much to be considered as like electron donating group. It does not modify the overall electron density that could affect the molecule's biological activity, pharmacological activity. It's not that significant. That's why we don't actually consider this to be an electron donating group that could change the electron density around the molecule, like the main reactive site of the molecule. There's, though in a sense it's electron donating group, we don't consider that part to be its main role. And yeah, we discussed why that's metabolic blocker. Next question was cyclophosphamide is commonly used in anti-cancer therapy. The structure was given, and they had asked which of the following statement is not true of the above structure. It is relatively non-toxic, it acts as a prodrug, it cannot be taken orally. Now, fourth, the structure is metabolized to release acrolein. So, the correct answer, I mean, the one that's not true, is that it cannot be taken orally. Cyclophosphamides are well known alkylating agents that are used in cancer therapy. Uh, you know, we'll discuss the following things about it. Cyclophosphamide is a nitrogen mustard derivative that functions as a prodrug. We discussed this, remember, even in last few slides. It remains inactive and it's in its administered form because it's a prodrug, and it is converted into a drug by hepatic metabolism, mainly by cytochrome P450, and that's how it gets converted from a prodrug to an active drug. It's widely used in treatment of lymphomas, leukemia, and solid tumors. So the first one, it says that it's relatively non-toxic. That's true because cyclophosphamide itself is relatively non-toxic because it's a prodrug. That's how you make a prodrug. The active metabolite, that is phosphoramide mustard, are responsible for its cytotoxic effect on rapidly dividing cancer cell. The toxicity occurs due to its metabolite, but the parent drug itself is less toxic. So yeah, that's why this was true. The second one, it acts as a prodrug. That's true because it is a prodrug that gets activated to its active drug form by liver enzymes like cytochrome P450, CYP. These are the name of the enzymes: CYP2B6 and CYP3A4 enzyme. They convert cyclophosphamide to 4-hydroxycyclophosphamide, which is then converted into phosphoramide mustard. That's the active alkylating agent. So this statement is also true. The third statement, the structure is metabolized to release acrolein. This is also true because this is the toxic byproduct of cyclophosphamide, and it causes bladder. It damages the bladder lining and causes hemorrhagic cystitis. So Mensa is often co-administered to neutralize acrolein and to protect the bladder. The fourth one was it cannot be taken orally. That's false because it is actually taken orally, and it's available in form of tablets as well as intravenous injections. Oral cyclophosphamides undergo first pass metabolism in liver because they are orally taken, which is then converted into active enzymes in the liver. The bioavailability of oral cyclophosphamide is high, making it effective when taken by mouth. That's why this statement was false, because it said that it cannot be taken orally. Hence, the right answer. The next question, L791456, the structure is given there, is an anti-arthritic drug. Which of the following structure is not a likely metabolite? So A, B, C, D. These were the four options. You had to say which one cannot be a likely metabolite. Like, this is not how this can be metabolized. So the correct option was A. This is not. There is no way this anti-arthritic drug L791456 is going to be metabolized into structure A. It can be metabolized into B, C, D, we'll discuss that, but cannot be metabolized into A. This is how. So it's an anti-arthritic drug. It has chlorinated pyridine ring, methyl sulfonyl group, SOMe, and then bipyridyl scaffold. That means it has two pyridine rings. It can undergo metabolism via phase 1 or phase 2 reactions, basically catalyzed by liver enzymes. So the common metabolic pathway would be oxidation, reduction, carboxylation, and conjugation. Conjugation, again, phase 2 metabolic pathway. So in oxidation, there can be hydroxylation, that is addition of OH, and N-oxidation, oxidation of nitrogen containing compound. Reduction, again phase 1, is nitro reduction if there is any nitro group present. Carboxylation, that's formation of carboxylic acid, and conjugation is basically sulfation or glucuronidation. For structure A, it contains OH group on pyridine ring, which is not a common metabolic pathway for such structure. Pyridine ring generally undergoes an oxidation reaction, but direct hydroxylation is uncommon. So that's highly unlikely. Why is it unlikely? Because pyridine is basically electron deficient. It makes them less susceptible for hydroxylation, and the metabolism this kind of structure, oxidation at nitrogen is preferred, or many alkyl group, and not directly to the aromatic ring. So that's why one cannot be the correct answer. In structure B, the features, basically it has, you know NO group. It shows oxidation there. So oxidation at nitrogen in heterocyclic rings often leads to an oxidation or nitro formation. So that makes more sense, and hence that can be a metabolite. Again, structure C, you can see there's this again. So it contains N oxide functional group on pyridine ring, which is again more likely than, so because it's a heteroatom, it's easier to oxidize the N there rather than the aromatic ring. Structure D has carboxylic group, you can see here, which is common metabolic oxidation product. Oxidation of methyl or nitrogen group can often lead to carboxylation, and we discussed this in previous slides as well. It's easier to metabolize the nitrogen or alkyl groups in such rings, in pyridine based structures, rather than you know um, do any kind of modification in the electron deficient site, which is basically the aromatic ring. So hence structure A was least likely to be any kind of metabolite for that kind of drug. Question 8. Why should the addition of an alcohol or phenol group to a drug decrease the drug's duration of action? 1. It acts as a polar handle for conjugation reaction. The conjugates are excreted more quickly. 2. It increases the polarity of a drug and reduces the amount of drug absorbed. 3. It reacts with proteins in the body such that the drug is irreversibly linked to the proteins by a covalent bond. Or 4th, it acts as an electron withdrawing group and affects the binding strength of important binding groups. The correct answer was it acts as a polar handle for conjugation reactions. The conjugates are excreted more quickly. So that's the whole point of metabolism part 1, part 2. So we'll see in metabolism part 1, they the drug is modified by oxidation, reduction, and hydrolysis. This introduces and exposes more polar functional group, like addition of hydroxyl, amine, things like that. In phase 2 metabolism, conjugation is done in such a way that more highly polar groups are attached to the drug. This increases water solubility of the drug, leading to rapid excretion via renal or via kidney biliary excretion. Once it becomes more soluble, it's easier to get filtered out. The role of alcohol and phenol as polar handles for conjugation. Well, the OH group in alcohol or phenol serves as recognition site for conjugation reaction. The body eliminates this such modified drugs through urine or bile. A common pathway would be like one, glucuronidation, the enzyme UDP, gluco gluconylyl transferase, that's called UGT. So what it does is basically it attaches OH group, making the drug more soluble. So morphine glucuronidation enhances the excretion in urine. Sulfation, it's common in phenols. So the enzyme used there is sulfotransferases. It's SULT. So they do that by adding sulfate group to phenol, and that makes the drug more polar and easy to excrete. The example is paracetamol. It undergoes sulfation, leading to easier elimination. Number three is methylation, and that's very less common compared to the other two, but it's still relevant, obviously. Um, you saw that in case of um, the drug where they introduce megasterol acetate, where they actually added that methyl group there. So addition of methyl group to phenol can basically reduce its activity and aid in excretion. And epinephrine is metabolized via methylation before elimination. So why does this decrease drugs duration of action? Well, conjugated drugs lose pharmacological activity and they cannot bind effectively to the targets. The drug is rapidly excreted, reducing the time it is active in the body, and that means the drugs with hydroxyl and phenol group are often short acting unless modified to prevent the metabolism. Why the other options were incorrect? Well, it stated one of the options stated that it increases polarity of the drug and reduces the amount of drug absorbed. Well, as we discussed, it is true, but not the main reason. Hydroxyl group after the addition can basically change the polarity, and that would lead to difference in how it's being absorbed. But absorption is not entirely dependent on one factor. Mm, it has various other factors, like just addition of one thing is not going to change the absorption entirely in most of the cases. So lipophilicity and transporters, if transporters are there, they can be you know easily brought inside the cell and taken out of the cell. Many drugs with hydroxyl groups are still well absorbed, especially if they use active transport mechanism, because then they're basically having an aid to bring things inside the cell, right? The other option stated it reacts with protein in the body such that the drug is irreversibly linked to the protein by a covalent bond. No, alcohols and phenols do not covalently bind to protein under normal physiological conditions. Covalent bonding is more characteristic of reactive electrophiles like alkylating agents in chemotherapy. The last one was it acts as electron withdrawing group and affect the binding strength of important binding groups. No, actually, OH is electron donating group rather than electron withdrawing group, and the main effect is by increasing hydrogen bonding potential. Because it is OH, and it does not alter the electron density at the binding site. So the main activity OH, it gets first involved with hydrogen bonding rather than its electron pair being involved in resonance. So here for a few examples of morphine versus codeine. Morphine, that contains hydroxyl group, undergoes rapid glucuronidation, leading to faster excretion. Codeine, which is methylated form of morphine, is more resistant to metabolism and has longer half-life. That's true. The next example is paracetamol. So in this group, it basically contains phenol group that makes it prime target for sulfation and glucuronidation. It is again rapidly metabolized and eliminated, and that explains why its half life is so less. The phenol group in salicylic acid, which is again a form of aspirin, salicylic acid undergoes glucuronidation that contributes to its shorter half life. So, yeah, presence of alcohol and phenol group in a drug provides a polar handle for conjugation reaction, making the drug more water soluble and rapidly excreted. These leads to shorter duration of action as a drug is removed from circulation before it can exert prolonged effects. The next question was methicillin is an important penicillin which was effective in 1960s against penicillin G resistant strains of S. aureus. Although the drug is more effective than penicillin G against resistant strain, it is not as active against strains which are susceptible to penicillin G. The methoxy substituent, shown in blue here, of the aromatic ring play an important role in drugs' effectiveness against penicillin G resistant strains. Which of the following statement is most likely the explanation? So the first one says the methyl group acts as a steric shield to protect aromatic ring from oxidation by metabolic enzymes in the body. Number two, the methyl group acts as steric shields to steric shields to protect the beta lactam ring from hydrolysis by enzymes produced by resistant bacteria. The methyl group acts as conformational blocker to orient the aromatic ring out of the plane of the side chain amide group, allowing better binding interaction with the target enzyme. The last one is the methyl group increase the electron density of the aromatic ring, allowing better binding interaction with the target enzyme. The correct answer is the methyl group acts as steric shield to protect the beta lactam ring from hydrolysis by enzyme produced by resistant bacteria. So penicillin, those who the penicillin, the mode of action for this kind of bacteria is those that are resistant. They basically destroy they are called beta lactamase. They basically destroy the beta lactam ring, okay, of penicillin. By adding this OH group, this not OH group, by basically adding methyl to this group, by making that methyl ester, what happens is it acts as a steric shield that basically protects the beta lactamase ring. They create little bit of steric hindrance, and the enzymes can no longer identify the beta lactam ring, and they don't hydrolyze the beta lactam ring. That's how they act. So conformational blocker and steric modification in drug design. I've made a small slide for this. So conformational blockers are basically, structural modification introduced into a drug molecule to restrict flexibility and to stabilize particular bioactive conformation and to improve its interaction with target receptor or enzyme. Its purpose is to enhance binding affinity to reduce off targets effect, and improve metabolic stability, and optimize pharmacokinetics. Okay, the methods are cyclization of molecule, they can be cyclized, introduction of the bulkier group, or use of bioesters, or basically introduction of double bond or ring constraint. That's how things are conformationally blocked. In steric shield modification in drug design, they basically add bulkier groups starting from methyl, which is smaller, and then increasing to ethyl, fluorine, tertiary butyl to block enzymes. So the example is fexofenadine. It's a non-sedating antihistamine. It has steric shield that prevents it from crossing the blood-brain barrier and hence reducing the sedation. So we've discussed example of steric shield and what they are. So yeah, that's how the the modification works. I have included things about fluorination and examples of it. So if you'd like to go through it, it will still be good. And this is comparison of the conformational blockers and steric shielding. So they do both have their own purpose and effect on metabolism and what kind of structural modification they are, and few examples. And the last question was, what sort of therapy involves administration of an antibody linked to an enzyme such that the enzyme activates a prodrug? The four options were given, out of which the correct answer was the third one, ADEPT, adapt. So I will discuss this later in the next slide because I'm running out of time. I have included few questions from this one, and for our next session, and I think I'll discuss that in the week 11, in the next session, which is week 11. Yeah, but at this point I hope this helps, and yeah, I think I'll conclude this, and we'll discuss everything else in our next session.
Okay, ma'am.
Yeah.
I think, like, can you take the fifth question, ma'am?
Yeah. Which one? Fifth question, right?
Yes, ma'am. That is, it is about the drug which, this no, fifth question.
Yeah.
This one.
Yeah.
For this, you said that it cannot be taken orally.
No.
But the thing is, we might not know what are the drugs that are currently available in tablet form or in the other form. But while referring the previous slide, you said that one of us metabolite that is a toxic thing.
Yeah, acrolein.
But uh, how we can conclude that, ma'am? Because we may not be sure of deciding that this drug can be taken orally or not, in what form it is available or not. But how can we decide over this?
Yeah, so actually this drug is taken orally, because it's a prodrug. It is taken orally and it undergoes first pass metabolism where it is converted into its active form. Uh, Once we get the drug, the first thing we do is formulation. Everything after getting the activity and the synthesis of drug, our main target is to determine the mode of administration. Our first target is always to make the drug more easier to administer, to get easier form of administration, and that's always, so oral administration is always preferred. So we study the way study the basically metabolism with different enzymes, right? If they are reaching their site of action and they are still bioavailable and cause less interaction with less side cause less side effect and have less interaction with all the enzymes, we always prefer the drug to be in the tablet form so that they are easily taken. But sometimes the drugs have issues like solubility, and so um, they are more uh, difficult to you know uh, how to say, they are difficult to be taken, they are difficult to convert. Not exactly, but there are various studies done for their formulation, you know like if it's more beneficial to be injected intravenously. And that's why we read the routes of administration, and we studied how different kind of drugs are formulated in a way that they can be more bioavailable, have less toxicity and everything. So it's part of their formulation. It takes a long time to come up with a good formulation for a drug that is less toxic and everything. You cannot just mm, guess that by looking at the structure of the molecule. You can have a well informed guess if you're very used to, if you are in the field of drug designing. So you might have some idea like, oh, these active uh, functional groups are quite exposed and they can lead to enzymatic hydrolysis, and that might not be very um, that can lead to toxic metabolites. So this cannot be taken orally. You can make a well informed guess based on your uh, basically information, your well registered information, and based on literature survey or something like that. But most of the time that's not very accurate, and you have to uh, read through things and do the experiments with different enzyme, read about their metabolites and everything, and then do the formulation. Am I able to clear that, Kritika? Is it like more clear now?
Oh my god.
Is
Phoning?
okay, Kritika. So if we're done, uh, do you have any other questions?
Sorry, ma'am. Actually, there was an interruption in my
No, no, it's
network,
okay.
so I couldn't unmute myself.
No, no, it's okay. It's okay. It's totally okay. So, is everything clear now?
Yes, ma'am. Thank you.
Okay. Okay, then I'll conclude the session. Bye.